One eerie night, a robber sneaks into a gas station with sinister intentions. He attempts to hijack an unattended car. Much to his dismay, he discovers that the car does not have enough fuel for a getaway. He is now faced with a dilemma that threatens to jeopardize the night: does the engine run on petrol, diesel, or natural gas? He needs to fill her up, and fast, before the owner gets back. He tries them one by one with the engine running, and finds that certain fuels when added are not used up. But petroleum, when added, disappeared immediately. So, he figured the engine runs on petrol, and made away just in time. When scientists of the early 20th century were confronted with an engine many times more complex, they adopted a similar experimental approach as the robber to decipher how it works. This engine was the animal muscle, and they were interested in knowing how living tissue converts food that we eat into energy for various activities. If this energy came from sugar, then it needed to be addressed how a complex carbohydrate molecule could break down into simpler substances during muscle respiration. Research into winemaking had led to the discovery of a pathway for the breakdown of glucose. Brewers had discovered that adding yeast press juice to grape sugars converted it to wine, with the liberation of carbon dioxide. When yeast juice was filtered through a cloth, the residue was by itself not sufficient to induce fermentation. Meyerhoff found that when muscle filtrate was added to yeast residue, fermentation could proceed. The presence in muscle tissue of a factor for fermentation suggested that fermentative reactions might take place in the muscle tissue as well. Thus, it was proposed that during respiration, muscle tissue first converts glucose to pyruvate, according to the pathway known from fermentation. Otto Warburg invented a device which he called the respirometer to measure gas usage by the muscle. It consisted of a fixed-volume glass jar, which was filled with crushed or ground muscle. There was a sidearm, which contained a substrate to be added to the muscle preparation. When the sidearm containing glucose was tipped into the main flask, respiration would begin. The process of respiration consumed oxygen in the closed flask, leading to a drop in pressure. The volume of oxygen used was then measured precisely using the rise or fall of mercury in the attached manometer. Warburg found that muscle respiration was an oxidative process, meaning that it required a net usage of oxygen from the atmosphere. As fermentation was anaerobic, while muscle respiration proceeded with the consumption of molecular oxygen, it became evident that the production of pyruvate was not the last step of respiration. Pyruvate was being further oxidized to carbon dioxide and water, but the identity of the intermediate molecules remained a mystery. Thinking like the robber, Torsten Thunberg reasoned that if any compound is an intermediate in muscle respiration, it would disappear immediately upon adding to muscle juice, because the muscle would use it as fuel accompanied by a consumption of oxygen. But if a compound is added, and no uptake in oxygen gas is seen, then it implies the compound is not being oxidized and so it is not an intermediate in the oxidation of pyruvate. With this general principle, Thunberg screened for a number of organic acids that were found in nature. He placed the salt of a different acid in the sidearm for each experiment and noted whether there was any change in the manometer reading suggesting oxidation of that acid by the muscle preparation. Among the many acids screened, he got a positive hit on some of them. Citric acid, derived from the juice of lemons and cucumbers, induced a strong increase in oxygen uptake. So did succinic acid, distilled from amber. Fumaric acid, from the fumaria plant. Malic acid, found in apples. Lactic acid, in sour milk, glutamate, an amino acid. As these acids were naturally occurring, they had been studied for decades and ways were available to assay each of them, that is, to chemically determine their presence in a solution. Additionally, Thunberg found that oxalic acid, which is found in abundance in the juice of sorrel, strongly decreased gas metabolism. It seemed likely to Thunberg that these acids were intermediates in the oxidation of pyruvate to carbon dioxide. But the order in which they were converted remained to be determined. Warburg proposed that the acids were being oxidized by a super enzyme present in the muscle, called the respiration enzyme, which oxidized all the added acids. Judah Hirsch-Quastel studied the oxidation of succinate to see if this was the case. First he took a muscle suspension and added succinic acid to it. As expected, the succinate was oxidized rapidly. Then, he screened for a number of poisons, that is, compounds which had the capacity to inhibit the oxidation of succinic acid. He found that in the presence of malonate, there was no uptake of oxygen by muscle tissue containing added succinate. Presumably, the target molecule of malonate was Warburg's respiratory enzyme. The absence of this oxidase would then prevent oxidation of not just succinate, but all the organic acids. To test this, Quastel experimented to see if malonate also inhibits the oxidation of fumaric acid. He found that adding malonate to muscle homogenate containing fumaric acid did not affect its rate of oxidation. So, here was evidence that the succinate enzyme was not the same as the fumarate enzyme. Further, Quastel took a suspension of muscle and added to it a saturating concentration of succinate, measuring the rate of oxidation in the manometer. Now, if the oxidase for glucose is the same as for succinate, then addition of succinate would not lead to any additional oxygen uptake, as the enzyme is already saturated with glucose. However, Quastel found that upon adding succinate and glucose, the rate of oxidation was the sum of the individual rates, suggesting a different oxidase for each acid. The disproving of Warburg's single enzyme view by Quastel opened the hunt for each of the individual enzymes. Using fractionation techniques, scientists attempted to purify muscle, liver, and vegetable preparations to isolate pure solutions which had the capacity to oxidize exactly one of the acids. Working with purified muscle extract, Batelli and Stern found that addition of succinic acid leads to the incorporation of oxygen by the animal tissue. They were able to isolate malic acid, as a product of oxidation, from the reaction mixture. Thus, they conjectured that malate is downstream of succinate, formed by the addition of an extra oxygen atom. Einbeck showed later that fumarate is the primary product and that malate is formed by secondary hydration of the fumarate. The enzyme fumarase was isolated from animals, plants, and bacteria, which catalyzed the reversible reaction fumarate to malate. The interconversion of the two acids was found to be very rapid compared to their formation or decomposition, at least in animal tissues, and the acids are therefore encountered in their equilibrium mixture. Further, based on known reaction pathways, it had long been expected that the biological oxidation of fumarate and malate would yield oxaloacetate. This was confirmed experimentally, and it was shown that fumarate is not oxidized directly to oxaloacetate, but only after conversion to malate. Albert Szent-Györgyi then performed an experiment in which he added oxaloacetate directly to muscle tissue suspension. Surprisingly, he discovered that within minutes, malate and fumarate were formed from the added oxaloacetic acid. This was a reductive reaction, which took place even under aerobic conditions. The reaction malate to oxaloacetate was thus reversible. Although many oxidations are reversible under some conditions, the rate of reduction is usually slow or negligible under the conditions prevailing in respiring muscle, that is, slow in the presence of oxygen gas. The rapid reduction of oxaloacetate in the presence of oxygen therefore indicated a special significance of the reversible system malate to oxaloacetate. Having studied the oxidation of acids in the succinate series, scientists then turned their attention to the oxidation of citric acid. From cucumber seeds, Wagner-Jauregg isolated an enzyme preparation that was capable of oxidizing citric acid and not any of the other acids found by Thunberg. He called it citric dehydrogenase. But in a random screen of this enzyme with other acids, he found that his enzyme was capable of oxidizing isocitric acid just as well as citric acid. Wagner-Jauregg was unable to identify the product of oxidation in both these cases, and the reaction mechanism of citric acid oxidation remained unknown. Franz Knoop, working with his student Karl Martius, studied the chemical structure of citric acid to propose a mechanism of oxidation on paper. They noted that citric acid has a central carbon atom, which was exposed to the effect of the two terminal carboxyl groups, and thus open to attack by oxidation. Martius and Knoop realized that the structure of citric acid could be imagined as being a molecule of malic acid, with an extra acetic acid substitution. They applied the known transformations of malic acid to this compound. First, a dehydration step to convert malic acid to fumaric acid yielded the compound cis-aconitic acid from citric acid. This is rehydrated to isocitric acid, with the hydroxyl group now being at the alpha carbon. The oxidation of malic acid to oxaloacetic acid can now be effected. This results in the formation of a beta-carboxylate alpha-ketoglutaric acid. Then, carbon dioxide can be split off to form alpha-ketoglutaric acid. The alpha-ketoglutarate was then proposed to be oxidized by conversion to succinate. In this manner, Martius and Knoop worked out a possible reaction mechanism for the degradation of citric acid using reactions that were already found to occur physiologically. Martius even found experimentally the equilibrium concentrations of the citric-isocitric acid system. As confirmation of their theory, Martius and Knoop added 1 gram of citric acid to an extract of liver enzymes, and succeeded in isolating 200 mg of alpha-ketoglutaric acid formed from it. They stated, one could scarcely find another pathway to explain the formation of the acid isolated here, and it does give firm support to the pathway outlined. Then, almost poetically, Martius and Knoop made the keen observation, we have repeatedly pointed out the great significance of physiological reversibility in biochemical reactions. That is why it strikes us as an interesting fact, that among the intermediaries appearing on this pathway, there are two acids that, according to our last report, so easily condense to citric acid, oxaloacetic acid and pyruvic acid. Indeed, Martius and Knoop had previously found that a mixture of oxaloacetate and pyruvate, dissolved in aqueous sodium carbonate in a test tube, formed citrate when treated with hydrogen peroxide. This reaction did not require any muscle extract, and thus it was quite conceivable that such a condensation could take place naturally in the oxidative environment of the muscle with the help of enzymes. The name aconitase was suggested for the enzyme or group of enzymes responsible for the interconversion of citric, aconitic, and isocitric acids. The action of aconitase, the reversible hydration of a double bond, was so similar to that of fumarase, that scientists wondered whether fumarase and aconitase were the same enzyme. But the discovery of specific inhibitors proved that they were separate entities. Extracts from the leaf of medlar were found to hydrate cis-aconitate but not fumarate. Further, fluoride was found to inhibit aconitase, but not fumarase, while monoiodoacetate inhibited fumarase but not aconitase. These findings indicated that fumarase and aconitase are different enzymes. The presence of a number of enzymes for the oxidation of 4-carbon di- and tri-carboxylic acids had been discovered in animal muscle. These were assumed to be intermediates in the oxidation of carbohydrate by the muscle, and thus directly acting as fuel to the muscle. But, Albert Szent-Györgyi would next make a discovery that turned this view upside down. He carried out careful volumetric measurements on the volume of oxygen consumed during the oxidation of the dicarboxylic acids, and the corresponding volume of carbon dioxide produced as dissolved bicarbonate, and noticed a glaring anomaly. The amount of oxygen consumed was far greater than could be accounted for by the oxidation of the acid alone. For example, if 1 mole of oxygen was required for the complete oxidation of succinate, then Szent-Györgyi instead found 6 moles of oxygen were consumed for that same amount of succinate. So, where was the extra oxygen going? Szent-Györgyi discovered that in addition to the dicarboxylic acids, if he added pyruvate to the muscle suspension, oxygen consumption was greatly stimulated. This was also the case if glucose, or any carbohydrate derivative from the fermentative pathway was added. Therefore, he concluded that the extra oxygen was likely being used for oxidation of carbohydrate. On the basis of these observations, Szent-Györgyi suggested that the main function of malate, fumarate, and oxaloacetate in muscle is not to serve as a fuel, but rather as a catalyst of endogenous substrates. In 1937, Hans Adolf Krebs, a student of Warburg, studied the two reversible systems of acids that were known to be rapidly oxidized in animal muscle. One was the reversible system of dicarboxylic acid succinate to fumarate to malate to oxaloacetate. The other was the reversible system of tricarboxylic acids citric to cis-aconitic to isocitric to alpha-ketoglutaric acid. Alpha-ketoglutarate was already known to undergo dehydrogenation to succinate, thus completing the link between the di- and tricarboxylic acid systems, and a linear chain of transformations was established. However, the fate of oxalic acid was not known. Further, these reactions were only shown to occur with isolated systems of enzymes. It still needed to be demonstrated that the reaction rates were rapid enough to occur in animal muscle generally. Due to the catalytic nature of the reactions, this was tricky to prove as the reactants were regenerated from the products in muscle, rendering it indistinguishable whether any reaction had taken place, with the only evidence being the high rate of oxygen uptake by respiring tissue. Krebs used pigeon breast muscle for his studies. This was the chief muscle employed in flying, and therefore one of the most powerful muscles in the animal kingdom. Krebs found it to be especially suitable for the study of oxidative reactions because this tissue maintains its oxidative capacity well after disintegration and suspension in aqueous media. He minced the muscle tissue and added some citrate to it, and measured the volume of oxygen absorbed after a fixed period of time. He found that without added citrate, the muscle absorbed 1 microliter of oxygen, but with citrate, it absorbed 2 microliter. The extra microliter of oxygen was far greater than could be accounted for by the oxidation of citrate alone. Therefore, Krebs reasoned that citrate had a catalytic effect for the oxidation of some other substrate, just like Szent-Györgyi's discovery with acids of the succinate series. To confirm this, Krebs took muscle suspension, and added to it both citrate, and also glycerophosphate, a carbohydrate derivative from the fermentative series. The oxygen consumption was far higher than if either citrate or glycerophosphate was added alone. This was good evidence that citrate was catalytically promoting the oxidation of carbohydrate, without being oxidized itself. In the catalytic process, citric acid was probably removed by a primary reaction, but regenerated by a subsequent reaction. In the balance sheet no citrate disappeared and no intermediate products accumulated. Therefore, Krebs first set out to find a substance that could cause added citrate to disappear from muscle suspension, because if the intermediate stages in the breakdown of citrate are known, then the mechanism of the catalytic effect will be clear. Krebs found that some poisons, like arsenite and malonate, bring about the effect of causing citrate to disappear. These poisons leave the breakdown of citric acid unaffected whilst they check the synthesis of citric acid. Krebs noted that in the presence of arsenite, only one molecule of oxygen is absorbed for each molecule of citric acid removed, which was not sufficient for the complete oxidation of citric acid. Therefore, one or more intermediates must accumulate. Krebs tested the resulting solution for the presence of any of the acids downstream in the breakdown of citrate. He found a volume of alpha-ketoglutarate equal to that of the added citrate. This complete yield suggested that arsenite is a specific inhibitor for the further oxidation of alpha-ketoglutarate, and that all the citric acid is converted to it. Similarly, Krebs found that in the presence of malonate, the oxidation of citric acid is checked at the stage of succinic acid. When citric acid and malonate were added together to minced pigeon muscle, succinic acid was detected in the solution, whereas only citric acid was detected if malonate was not present. Malonate, therefore, inhibited the oxidation of succinic acid. Krebs now had established a linear series of reactions that described the breakdown of citrate in animal tissue in the presence of oxygen. If it is true that the oxidation of citric acid is a stage in the catalytic action of citric acid, then it follows that citric acid must be regenerated eventually from one of the products of oxidation. Krebs next examined whether citric acid can be resynthesized from any of the intermediates of the citric acid breakdown, under non-oxidative conditions. For this, he flushed the holding flask of the Warburg apparatus with nitrogen gas to dispel all oxygen, then started a reaction in this anaerobic environment. Krebs was familiar with the test tube experiments of Martius and Knoop, where citrate was generated from oxaloacetate and pyruvate, so he decided to test whether such a reaction could take place in the animal tissue anaerobically. He added oxaloacetate to minced muscle suspension, and incubated it for 20 minutes in a water bath. After this, the solution was tested for the presence of citric acid. Citric acid was detected but the yield was comparatively low, amounting to no more than a few % of the added oxaloacetate. Citrate accumulated only if very high amounts of oxaloacetate were used. Krebs attempted to justify this by saying that the citrate which forms from oxaloacetate is rapidly broken down to alpha-ketoglutarate. He showed that the yield of citrate and alpha-ketoglutarate taken together was 25% of the added oxaloacetate, and that this was the maximum yield that could be expected, as the rest of the oxaloacetate was reduced to malate under the experimental conditions which were anaerobic. All the other acids tested by Krebs, including pyruvic acid, did not produce any citric acid under the same conditions. This experiment showed that muscle is capable of forming large quantities of citric acid if oxaloacetate is present, and the question arises, from which substance the two additional carbon atoms of the citric acid molecule are derived. Since the catalytic effect of citrate was more pronounced if carbohydrate molecules were present, it appeared that the substance condensing with oxaloacetate was derived from carbohydrate, which Krebs called triose. But due to the low yield of citric acid, there was still doubt about the proposed cyclical nature of citric acid breakdown. Therefore, Krebs undertook an experiment to show that a cycle does in fact exist. It will be recalled that malonate inhibits specifically the reaction, succinic acid to fumaric acid. Under reductive conditions, that is, in the absence of oxygen, oxaloacetate is reduced to succinate via the reversible system oxaloacetate to malate to fumarate to succinate. However, if the inhibitor malonate is also added, then no succinate should be expected to form from added oxaloacetate, and Krebs found that this was indeed the case. However, when the same experiment was carried out in the presence of oxygen, where oxaloacetate was added to muscle suspension, large amounts of succinic acid accumulated. As malonate was still present in the experimental conditions blocking the reductive transformations, it followed that succinic acid had formed from oxaloacetate via an oxidative route, with malonate preventing the secondary breakdown of succinate, once it had formed. So, it was shown that there were two pathways leading from oxaloacetate to succinate. A direct reduction through malate, and an oxidative reaction through citrate. Together, they formed the citric acid cycle.
In order to test this theory, we are making two preparations of the same voluntary muscle. From the first preparation, we are extracting all the chemical compounds of the muscle except the filaments. The muscle juice contains a number of chemicals, including the compound adenosine triphosphate.